Unit 3 Cell Structure and Functions. Chapter 8 Cell the Unit of Life. Chapter 9 Biomolecules. Chapter 10 Cell Cycle and Cell Division. Biology is the study of living organisms. The detailed description of their forms and appearance only brought out their diversity. It is the cell theory that emphasized the unity underlying this diversity of forms, that is, the cellular organization of all the life forms. The description of the cell structure and the cell growth by division is given in the chapters comprising this unit. Cell theory also created a sense of mystery around the living phenomena, that is, physiological and behavioral processes. This mystery was the requirement of integrity of cellular organization for living phenomena to be demonstrated or observed. In studying and understanding the physiological and behavioral processes, one can take a physiochemical approach and use cell free systems to investigate. This approach enables us to describe the various processes in molecular terms. The approach is established by analysis of living tissues for elements and compounds. It will tell us what types of organic compounds are present in the living organisms. In the next stage, one can ask the question, what are these compounds doing inside a cell? And in what way they carry out gross physiological processes like digestion, excretion, memory, defense, recognition, etc. In other words, we answer the question, what is the molecular basis of all physiological processes? It can also explain the abnormal processes that occur during any diseased condition. This physicochemical approach to study and understand living organisms is called reductionist biology. The concepts and techniques of the physics and chemistry are applied to understand biology. In chapter 9 of this unit, a brief description of biomolecules is provided. G.N. Ramachandran, 1922 to 2001, an outstanding figure in the field of the protein structure, was the founder of the Madras School of Conformational Analysis of Biopolymers. His discovery of triple helical structure of collagen, published in Nature in 1954, and his analysis of the allowed conformations of proteins through the use of Ramachandran plot rank among the most outstanding contribution in the structural biology. He was born on October 8th, 1922, in a small town not far from Cochin on the southwestern coast of India. His father was a professor of mathematics at a local college and thus had a considerable influence in shaping Ramachandran's interest in mathematics. After completing his school years, Ramachandran graduated in 1942 as a top ranking student in the BSc Honours Physics course of the University of Madras. He received a PhD from Cambridge University in 1949. While at Cambridge, Ramachandran met Linus Pauling and was deeply influenced by his publications on the models of alpha helix and beta sheet structures that directed his attention to solving the structure of the collagen. He passed away at the age of 78 on April 7, 2001. Chapter 8 Cell, the Unit of Life. When you look around, you see both living and non living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have, which a living thing has? The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of single cells and are called unicellular organisms, while others, like us, composed of many cells and are called multicellular organisms. What is cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of cell does not ensure independent living. Hence, cell is the fundamental, structural, and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. Invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory in 1938, Matthias Claden, a German botanist, GB, examined a large number of plants and observed that all the plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodore Swan, 1839, and in 1838, Matthias Claden, a German botanist, and at the same time, uh, Theodore Swan, 1839, a British zoologist, BZ, a British zoologist, studied different types of animal cells and reported that the cells had thin outer layer which is today known as plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of the cell wall is a unique character of the plant cells. On the basis of this, Swan proposed the hypothesis that the bodies of the animals and the plants are composed of cells and products of cells. Scleden and Swan, Scleden and Swan together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow 1855 first explained that the cells divided and new cells are formed from the pre-existing cells omnis cellula e cellula. He modified the hypothesis of Scleden and Swan to give the cell theory a final shape. The cell theory, as understood today, all the living organisms are composed of cells and products of cells. All cells arise from the pre existing cells. An overview of the cell. 8.3. You have earlier observed cells in an onion peel and or a human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as a delimiting structure of the cell. Inside each cell is a dense membrane bound structure called the nucleus. This nucleus contains a chromosomes which in turn contain genetic material DNA. Cells that have the membrane bound nuclei are called eukaryotic, whereas the cells that lack membrane bound nucleus are called prokaryotic. In both prokaryotic and eukaryotic cells, a semi fluid matrix called cytoplasm occupies the volume of the cells. The cytoplasm is the main arena of cellular activity in both plants and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane bound distant structure called organelles like the endoplasmic reticulum, the Golgi complex. Beside the nucleus, eukaryotic cells have other membrane bound distinct structures called organelles like the endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, uh, microbodies, and vacuoles. The prokaryotic cells lack such membrane bound organelles. Ribosomes are non-membrane bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles chloroplast in plants and mitochondria and on rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelles called centrosome, which helps in the cell division. Cell differs greatly in size, shape, and activities. For example, mycoplasmas, the smallest cells, are only zero point 0.3 micrometer in length, while the bacteria could be 3 to 5 micrometer. Zero point 0.3 micrometer of about the mycoplasma, which is the smallest cells. Smallest cells are only zero point 0.3 micrometer in length, while the bacteria, ठीक है, the largest isolated cells in the egg is the egg of an ostrich. Among multicellular organisms, human blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc like, polygonal, columnar, cuboidal, thread like, and are irregular, even irregular. The shape of the cell may vary with the function they perform. Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, that is pneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of the bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, and spiral. The organism of the organization of the prokaryotic cells is fundamentally similar, even though prokaryotes exhibit the organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shape and functions. All prokaryotes have a cell wall. Sorry. All prokaryotes have a cell wall surrounding and the cell membrane except in mycoplasma. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by the nuclear membrane. In addition to the genomic DNA, the single chromosome or the circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistant to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. It's used to monitor bacterial transformation with the foreign DNA. Nuclear membrane is found in the eukaryotes. Now, no organelles like the ones in the eukaryotes are found in the prokaryotic cells except for the ribosomes. Prokaryotes have something unique in the form of the inclusions. A specialized differentiated form of the cell membrane called the mesosome is the characteristic of prokaryotes. Characteristics of prokaryotes: mesosome, differentiated form of the cell membrane called the mesosome, is the characteristic. They are essentially infoldings of cell membrane. Red blood cells round and biconcave. White blood cells amoeboid, columnar, and epithelial cells columnar. Epithelial cells long and narrow. Nerve cells branched and long. Tracheate elongated. Mesophyll cells round and oval. It is bacteria 10 to 20 micrometer, but it's here written bacteria. It was written somewhere here. 3 to 5 micrometer, while a bacteria could be a 3 to 5 micrometer. Here it is written typically eukaryotic cells. Eukaryotic cells. Bacteria is a prokaryotic cell. Typical bacteria 1 to 2 micrometer. Typical bacteria. PPLO pleuropneumonia like organisms, that is zero point 0.1 micrometer, and viruses that is 0.02 to zero point 0.2 micrometer. That is the smallest. No, that's smallest one is mycoplasma, zero point 0.3 micrometer. Cell envelope and its modification. Most cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of tightly bound three-layered structure: the outermost glycocalyx, followed by the cell wall, and then the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by the gram. That those that take up the gram stain are gram positive, and others that do not are called gram negative bacteria. Glycocalyx differ in the composition and the thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it can be a thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. Okay, it is a glycocalyx differs in the composition and thickness. If it is a loose sheet, that it is a slime layer, and some, while in other it may be thick and tough called the capsule. The cell wall different determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. It This membrane is similarly structurally structurally to that of the eukaryotes. Okay, first there it comes the glycocalyx, followed by the cell wall, and then the plasma membrane, which is the selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is called the mesosome, which is formed by the extension of the plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. Vesicles, tubules, and lamellae, they help the cell wall formation, DNA replication, and distribution to the daughter cells. They also help in respiration secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions like into the cytoplasm called the chromatophores, which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called flagella. Bacteria show a range in the number and the arrangement of the flagella. Bacterial flagellum is composed of three parts: filament, hook, and the basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made up of sessile protein. The fimbriae are small bristle-like structures, fibers sprouting out of the cells. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. Ribosomes and the inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cells. They are about 15 nanometer by 20 nanometer in size. 15 nanometer by 20 nanometer in size and are made up of two subunits 50s and 30s, which present together for from 70s prokaryotic ribosomes. Ribosomes are the site of the protein synthesis. Okay, they are about 15 nanometer to 20 nanometer in size and they are made up of two subunits 50s and that is Swedborg units that are together called 70s prokaryotic ribosomes. Ribosomes are the site of the protein synthesis. Several ribosomes may attach to the single mRNA and form a chain called the polyribosomes or polysomes. The ribosomes of the polysome translate the mRNA into proteins. Inclusion bodies, reserve material in the prokaryotic cells, are stored in the cytoplasm in the form of the inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Inclusion bodies, the reserve material in the prokaryotic cells, are stored in the cytoplasm in the form of the inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm, that is, phosphate granules, synovial granules, and glycogen granules. Gas vacuoles are found in the blue, green, and purple and greens photosynthetic bacteria. Sorry. Eukaryotic cells. The eukaryotes includes all the protist eukaryotic cells. The eukaryotes includes all the protist plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of the cytoplasm through the presence of the membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different, as the former possess cell walls, plastids, and a large central vacuole which are absent in the animal cells. On the other hand, animal cells have centrioles which are absent in most of the all plant cells. Although eukaryotic cells are not identical, plants and animal cells are different as the former, that is the plant cells possess cell walls, plastids, and a large central vacuole which are absent in the animal cells. On the other hand, animal cells have centrioles which are absent in almost all plant cells. This is the diagram showing the plant cell and animal cells. This portion is the plant cell where the presence of smooth endoplasmic reticulum, lysosome, smooth endoplasmic reticulum, rough endoplasmic reticulum, plasmodesmata, mitochondria, peroxisome, cytoplasm, chloroplast, ribosomes, mitochondrion, cell wall, middle lamella, वैक्यूल प्लाज्मा मेम्ब्रेन सेल वॉल के बाद आता है प्लाज्मा मेम्ब्रेन सेल न्यूक्लियर मेम्ब्रेन न्यूक्लियर एनवेलप गोल्गी apparatus nucleolus nucleus and then here comes the animal cells here we can see the microvilli plasma membrane centriole peroxisome lysosome, ribosome, mitochondrion, rough endoplasmic reticulum, cytoplasm, nucleus, nucleolus, nuclear envelope, smooth endoplasmic reticulum, Golgi apparatus. Let us now look at the individual cell organelles to understand their structure and function. Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in 1950s. Meanwhile, chemical studies on the cell membrane, especially in the human red blood cells, enabled the scientists to deduce the possible structure of the plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. Lipids and proteins. The major lipids are phospholipids that are arranged in the bilayer. Also, the lipids are arranged within the membrane with the polar heads towards the outer side and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of the saturated hydrocarbon is protected from the aqueous environment. Non-polar tail was protected from the aqueous environment. ठीक है. In addition to the phospholipid membrane also contain cholesterol. Later, biochemical investigation clearly revealed that the cell membranes also possess protein and carbohydrate. The ratio of the protein and lipids varies considerably in different cell types. In human beings, this membrane of the erythrocytes has approximately 52% of the protein ज़्यादा होता है 52% of the protein and 40% of the lipids. Depending on the ease of the extraction, membrane proteins can be classified into integral and peripheral. Peripheral proteins lie on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. फोस्फोलिपिड्स का जो लेयर होता है उसमें लिपिड्स एंड प्रोटीन्स प्रेजेंट है. प्रोटीन्स ज़्यादा होते हैं 52%. So the lipids, the major lipids, the studies showed that the cell membrane is mainly composed of lipids, and some major lipids or phospholipids are arranged in the later the biochemical investigation clearly revealed the cell membrane also possess proteins and carbohydrates. Okay. Fluid mosaic model of the plasma membrane that has sugars and peripheral protein, integral protein which is buried, peripheral protein, integral protein, and phospholipid bilayer as we know already forming the also phospholipid bilayer has cholesterol in it inside the. And where the cholesterol present, let me read that. In addition to the phospholipids, membrane also contain cholesterol. An improved model of the structure of the cell membrane was proposed by Singer and Nicholson 1972, widely accepted as fluid mosaic model in the figure 8.4, which we already saw. According to this, the quasi-fluid nature of the lipid enables lateral movement of the proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of the view of the function like cell growth, formation of the intercellular junction, secretion, endocytosis, cell division, etc. According to the quasi-fluid of the lipid enables the lateral movement of the proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on the either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is high concentration to the lower concentration. Water may also move across this membrane from the higher concentration to the lower concentration. Movement of the water by the diffusion is called osmosis. As the polar molecules cannot pass through the non-polar bilipid layer, they require a carrier protein of the membrane. Carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is their lower to the higher concentration. Such transport of energy dependent is an energy dependent process in which ATP is utilized and it is called active transport. Example, sodium or potassium pump. Cell wall, as you may recall, a non living rigid structure called the cell wall, forms an outer covering of the plasma membrane of the fungi and plants. Cell wall not only gives shape to the cells and protects the cell from the mechanical damage and infection, it also helps in cell to cell interaction and provides barrier to the undesirable micromolecules. Algae have cell wall made up of cellulose, keratins, mannans. We are talking about algae and minerals like calcium carbonate, while in the other plants it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of the young plant is called primary wall, is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner side towards the membrane of, of the cell. The middle lamella is the layer mainly calcium pactate. Middle lamella is a layer which mainly calcium pactate which holds or glues the different neighboring cells together. The cell wall and middle lamella may be traversed by the plasmodesmata in which they connect the cytoplasm of the neighboring cells endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as the endomembrane system because of their function as the coordinate. The endomembrane system consists include the endoplasmic reticulum, Golgi complex, lysozyme. Sorry. In terms of the structure and function, many of these are considered together as the endomembrane system because of their function and our functions are coordinated. The endomembrane system include endoplasmic reticulum, Golgi complex, lysosome, Gerluvi complex, lysosomes, and vacuoles. Since the functions of the mitochondria, chloroplast, and peroxisomes are not coordinated with the above components, these are not considered as the endomembrane system. The endomembrane system first 8.5.3.1, the endoplasmic reticulum ER. The electron microscope studies of eukaryotic cells reveal the presence of a network of reticulum of tiny tubular structures scattered in the cytoplasm that is called endoplasmic reticulum. Hence, endoplasmic reticulum divides the intercellular space into two different compartments, that is luminal inside endoplasmic reticulum and extraluminal, that is outside the cytoplasm, which is outside the endoplasmic reticulum compartments. The endoplasmic reticulum often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on the surface is called rough endoplasmic reticulum. In the absence of the ribosome, they appear smooth and are called smooth endoplasmic reticulum. Rough endoplasmic reticulum is frequently observed in the cells actively involved in the protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. Extensive and continuous with the outer membrane of the nucleus, the smooth endoplasmic reticulum is the major site of the synthesis of the lipid. The animal cells lipid-like steroidal hormones are synthesized in the smooth endoplasmic reticulum. Golgi apparatus Camillo Golgi in 1898 first observed densely stained reticular structures near densely stained reticular structures near the nucleus. These are later named as Golgi apparatus after him. The Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae of zero point 0.5 micrometer to 1 micrometer diameter, and these are stacked parallel to each other. A varied number of the cisternae are present in the Golgi complex. The Golgi cisternae are concentrically arranged near the cells and with distinct convex cysts or forming convexes or the forming face and the concave trans. Concave trans are the maturing phase. The cyst and the trans phase of the organelles are entirely different but interconnected. Again, These are stacked parallel to each other. Varied number of cisternae are present in the Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus, which with distinct convex structure. Convex cis or the forming phase. Cis, where is cis phase? Cisternae, cis phase, ribosome. Let's see where it comes. Cis phase and the concave trans phase. Concave is just trans phase, which is the maturing phase. The cis and trans phase of the phases of the organelles are entirely different but interconnected. The Golgi apparatus principally perform the function of packaging materials to be delivered into either in the intracellular targets or the secreted outside the cells. Materials to be packaged in the form of vesicles from the ER fuse with the cis phase of the Golgi apparatus, okay, and move towards the maturing phase. This explains why Golgi apparatus remains in the close association with the endoplasmic reticulum. A number of the proteins synthesized by the ribosomes of the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from the transphase. Okay, are modified in the cisternae of the Golgi apparatus before they are released from the transphase. Golgi apparatus is an important site for the formation of the glycoproteins and the glycolipids. Lysosomes. These are membrane bound vesicular structures formed by the process of the packaging of the Golgi apparatus, formed by the Golgi apparatus, and the isolated structural vesicles have been found to be very rich in the almost all types of hydrolytic enzymes, hydrolases, lipases, lipases, proteases, carbohydrates, optimally active at the acidic pH. These pH are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. Vacuoles. The vacuole is the membrane-bound space found in the cytoplasm. This contains water, sap, excretory products, and other membranes are not useful for the cells. Other materials which are not useful for the cells, the vacuoles is bound by a single membrane called the tonoplast. In plant cells, the vacuoles can occupy only up to the 90% of the volume of the cells. In plants, in plant cells, 90% of the volume of the cell in plants, the tonoplast facilitates the transport of the number of the ions and other materials against the concentration gradients into the vacuoles. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. Sorry. In amoeba, the contractile vacuole is important for the osmoregulation and excretion. In many cells, as in the protist, food vacuoles are formed by engulfing the food particles. Mitochondria, single mitochondrion, unless specifically strained mitochondria are visible under the microscope. The number of the mitochondria per cell is variable depending on the physiological activity of the cells. In terms of shape and size, also considerable degree of variability is observed. Typically, it is a shaped or sausage shaped or cylindrical, having a diameter of zero point 0.2 to 1 micrometer, average zero point 0.5 micrometer, and the length of the 1 to 4.1 micrometer. Each mitochondria is a double membrane wall structure with the outer membrane and the inner membrane, dividing its lumen distinctly into two aqueous compartments, that is the outer compartments and the inner compartments. The inner compartment is filled with a dense homogeneous substance called matrix, and the outer forms the continuous limiting boundary of the organelles. The inner membrane forms a number of enfoldings called the cristae, singular crista towards the matrix. The cristae increases the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of the aerobic respiration. They produce a cellular energy in the form of ATP. Hence, they are called powerhouses of the cells. The matrix also possesses a single cellular circular DNA molecule. A few RNA molecule, ribosomes, 70S, and the components are required for the synthesis of the proteins and the mitochondria divided by the cells. Divided by the fission, plastids are found in all the cell plants and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus the imparting specific colors of the plants based on the type of the pigments. Plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoid pigments which are responsible for the trapping light energy essential for photosynthesis in the plastids are found in all the plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting the specific colors to the plants. Based on the type of the pigments, plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contains chlorophyll and the carotenoid pigments, which are responsible for the trapping of the light energy essential for photosynthesis. And in the chromoplast, fat soluble carotenoid pigments like carotene, xanthophylls, and others are present. This gives carotene, xanthophylls, and others are present. This gives the part of the plant in yellow, orange, or red color. The leucoplast are the colorless plastids of the varied plants and animals, varied shapes and sizes with the stored nutrients. Amyloplast stores carbohydrates. Amyloplast stores carbohydrate starch, that is potato, and alleoplast stores oils and fats, whereas the alleroplast contains stores proteins. Majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. If the chloroplast of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped oval, spherical, discoid, or even ribbon-like organelles having variable length, a 5 to 10 micrometer, and width is 2 to 4 micrometer. Their number varies from 1% of the chlamydomonas of a green alga to the 20 to 40% of the mesophyll. Lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micrometer and width is 2 to 4 micrometer. Their number varies 1% in the chlamydomonas or a green algae. Chlamydomonas, a green algae, to 20 to 40% in the mesophils. Like mitochondria, the chloroplast are also double membrane wound. One of the two, the inner chloroplast membrane, is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. The a number organized flattened membranous sacs called the thylakoids uh, present in the stroma. Thylakoids are arranged in the stacks like the piles of the coins are called grana or the integral thylakoids, intergranal thylakoids. In addition, there are flat membranous tubules called the stromal lamellae connecting the thylakoids of the different grana. The membrane of the thylakoids encloses a space called the lumen. Membrane of the enclosure space called a lumen. The stroma of the chloroplast contains enzyme required for the synthesis of the carbohydrates and proteins. It also contains small double membrane circular DNA molecules and ribosomes. Okay. Chlorophyll consists pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller, 70s, than the cytoplasmic ribosomes, 80s. Ribosomes are the granular structures first observed under the electron microscope as a dense particles by the George Ballard 1955. 1953. They are composed of ribonucleic acid RNA and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80s while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, larger and the smaller subunit. The two subunits of the 80s RNA are the 60s and 40s, while of the 70s is called 50s and 30s. That here S is the Swedborg's units stand for the sedimentation coefficient. It is directly measure of the density and the size. Both 70S and the 80S ribosomes are composed of two subunits, cytoplasm, cytoskeleton. An elaborate network of the filamentous proteinaceous structure consists of a microtubules, elaborate network of filamentous proteinaceous structure consisting of the microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as cytoplasm. Okay. Elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is called collectively referred to as cytoskeleton. The cytoskeleton in the cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Section of the cilia and flagella showing different parts. First one is the A is electron micrograph, which is the of the cilia or the flagella showing in the different parts, and B is the diagrammatic representation of the internal structure, which has plasma membrane, peripheral microtubules, which are doublets, inter-doublet bridge connecting two peripheral microtubules, doublets, and it has then central microtubule and then radial spoke and a central sheath. Cilia and flagella. Cilia and flagella are hair outgrowths of the cell membrane. Cilia are the small structure which work like an oars, causing the movement of the either of the cell or the surrounding fluid. Flagella are comparatively longer and responsible for the cell movement. The prokaryotic bacteria have also possess flagella, but these are structurally different from that of the eukaryotic flagella. Okay, the electron microscope studied of a cilium or a flagellum show that they are covered with the plasma membrane. Their core, called the exonym, possesses a number of microtubules running parallel to the long axis. Exonym. Okay, the exonym usually have nine doublets of the radially arranged peripheral microtubules and a pair of centrally located microtubules. Okay, such an arrangement of the exonymal microtubules is referred to as 9 plus 2 array. That is 9 plus 2 arrangement in the figure 8.10. 8.10, that is 9 plus 2, 1 2 3 4 5 6 7 8 9. 9 plus 2 arrangement of the 9 plus 2 array. The central tubules, which is it is present in the cilium or the flagellum, shows that are covered by the plasma. Their core is called exonym and possess a number of the microtubules. Okay, this is about the cilia or the flagellum. 9 plus 2 arrangement. The central tubules are connected by the bridges and is enclosed by the central sheaths, which is connected to one another. One of the tubules of the each peripheral doublets by the radial spoke. There are 9 radial spokes. Okay, the peripheral doublets are also interconnected by the linkers. Both cilium and flagellum emerge from the centriole-like structure called the basal bodies. Okay, centriole and centrosome and centriole. Centrosome is an organelle usually containing two cylindrical structures called the centrioles. Centrosome, which has two cylindrical structures called the centrioles. They are surrounded by the amorphous peripheral pericentriolar materials. Amorphous pericentriolar materials, they are surrounded by the amorphous pericentriolar materials. Both centrioles in the centrosomes lie perpendicular to each other, in which each has the organization like a cartwheel. They are made up of 9 evenly spaced peripheral fibrils of the tubulin protein. Each of the peripheral fibrils is called triplet. The adjacent triplet are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with the tubules of the peripheral triplets by the radial spokes made up of protein. The centrioles form the basal body of the cilia or flagella and the spindle fibers that give rise to the spindle apparatus during the okay. Centrioles form a basal body of the cilia or flagella. Okay, it's already written up there, the last sentence of that paragraph of cilium and flagellum, and central basal body of the flagella and the spindle fibers that give rise to the spindle apparatus during the cell division in the plant cells. Here, the peripheral doublets are interconnected by the linkers. Both the cilium and the flagellum emerge from the centriole-like structures called the basal bodies. Okay. Nucleus. Where is the structure? I can't see the structure, right? 9 plus 0 arrangement. Nucleus. Nucleus are the cell organelles are was first described by the Robert Brown as early as 1831. Later the material of the nucleus stained by basic dyes given the name chromatin by the Fleming. This is the structure, the figure given 8.11. Structure of the nucleus, nucleoplasm. We can see the nucleoplasm, nucleolus, and then the nuclear pore is present in the nuclear pore is present in the nuclear membrane. The interface nucleus. Nucleus of the cells, when it is not dividing, has highly extended and elaborate nucleoprotein fibers called the chromatin. Okay. Nuclear material, nuclear matrix, and one or more spherical bodies called the nuclei. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with the space between Microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nanometer called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. Okay, first say electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between okay, now I got it, space between 10 to 50 nanometer called the perinuclear space, forms the barrier between the materials present inside the nucleus and thus that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At the at the number of the places the nuclear envelope is interrupted by the minute pores. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of the two membranes. Okay, this these nuclear pores are passes through the which the movement of RNA or protein molecules takes place in both the directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variations in the number of the nuclei are also frequently observed. Can you recollect the names of the organisms that have more than one nucleus per cell? Some mature cells even lack nucleus, that is erythrocytes of many mammals and sieve tube cells of the vascular plants. Would you consider these cells as living? The nuclear matrix or the nucleoplasm consists of nucleolus and chromatin. The nuclei are spherical structure present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm as it is not membrane bound structure. It is the site for the active ribosomal RNA synthesis. Larger and more numerous nuclei are present in the cells actively carrying out protein synthesis. You may recall that the interphasal nucleus has a loose and a distinct network of nucleoprotein fibers called the chromatin. But during different stages of the cell division, cells so structured chromosomes in place of the nucleus chromatin contains DNA and some basic proteins called histones, some non-histone proteins, and also RNA. A single human cell has approximately 2 meter long thread of DNA distributed among its 43 pairs. 23 pairs, 46 chromosomes, it is 23 pairs. You will study the details of DNA packaging in the chromosome in class 12th. The every chromosomes visible only in the dividing cells. Every chromosomes essentially have a primary construction or a centromere on the sides of which disc shaped structures called kinetochores are present. Centromere holds two chromatids of the chromosomes based on the position of the centromere. Centromere holds two chromatids of the chromosomes based on the position of the centromere. Okay, the chromosomes are classified into four types. The metacentric chromosomes are the middle centromere forming the two equal arms of the chromosomes. The submetacentric has a centromere slightly away from the middle of the chromosomes, resulting in one shorter arm and the other one longer. And the in case of some metacentric acrocentric chromosomes, the centromere is situated close to its end, forming one extremely short and the one long arm, whereas the telocentric chromosome has the terminal centromere, telocentric long arm. But how it is denoted by LXLZZ. Sometimes a few chromosomes have non-staining chromosome secondary constructs. Sometimes a few chromosomes have non-staining secondary constructions at a constant location. This gives the appearance of small fragments called the satellite. Okay, microbodies meaning membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plants and animal cells. Summary: all organisms are made up of cells or aggregates of cells. Cells vary in their shape, size, and activities or functions based on the presence or absence of a membrane bound nucleus and other organelles. Cells and hence the organisms can be named as the eukaryotic or prokaryotic. A typical eukaryotic cell consists of a cell membrane, nucleus, and cytoplasm. Plant cells have a cell wall outside of the cell membrane. The plasma membrane is selectively permeable and facilitates the transfer of several molecules. The endomembrane cells includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. All the cell organelles perform different but the specific functions. Centrosomes and centriole form the basal body of the cilia and flagella that facilitate the locomotion. In animal cells, centrioles are also form the spindle apparatus during the cell division. Nucleus contains nuclei and chromatin network. It only not only controls the activity of the organelles but also plays a major role in the heredity. Endoplasmic reticulum contains the tubules of or the cisternae. They are of two types, rough and smooth. Endoplasmic reticulum helps in the transport of the substances, synthesis of the proteins, lipoproteins, and glycogen. The Golgi body is a membranous organelles composed of the flattened cells. The secretion of the cells that are packed in them and transported from the cells. Lysosomes are the single membrane bond containing enzyme for digestion of all the types of the macromolecules. Ribosomes are involved in the protein synthesis. These occur freely in the cytoplasm as in the associated with eth- endoplasmic reticulum. Mitochondria help in the oxidative phosphorylation and generation of the adenosine triphosphate, that is ATP. They are bound by the double membrane in the outer double membrane. They're bound by double membrane. The outer membrane is smooth and the inner membrane is the folds several cristae. The plastids are the pigment containing organelles found in the plant cells only. In the plant cells, chloroplasts are responsible for trapping light energy essential for the photosynthesis. The grana in the plastids is the site of the light reaction and the stroma is the site for the dark reaction. The green colored plastids are chloroplasts which contain chlorophyll, the whereas the other colored plastids are chromoplasts which may contain pigments like carotene and xanthophyll. Okay, chromoplast which may contain carotene and xanthophyll. The nucleus is enclosed by the nuclear membrane, nuclear envelope, or double membrane structures with nuclear pores. The inner membrane encloses the nucleoplasm and the chromatin material, and the cells is the structural and functional unit of life. Thank you.